Okay, so hello and welcome everyone to today's live class session of week 11 on the course Fundamentals of Protein Chemistry, which is being taught by Professor Shagota Dasgupta of IIT Kharagpur. And I am Srijan Sen, and today I will be taking a live class session on the courses or the lectures that were taught to you in this week and discussing with you in brief. So, and also we'll be discussing with you a few question and answers that will help you in solving the assignment and taking up the future exams that is coming up by the end of this month. Okay, so let us start with the lectures. I'm sharing my slide to you all. So basically in today's class we would be mostly focusing on protein-protein interactions. In the previous class we discussed about protein carbohydrates and protein nucleic acid and a few of the techniques regarding that on the identification of DNA and RNA. We talked about that. So in today's class we would be dealing with protein-protein interactions. So as from the name we can understand that protein-protein interactions occurs between two or more proteins that bind together. So when these two or more proteins binds together, they form an interface or some kind of depressions and cavities, which you can also call. So these proteins control and mediate many of the biological activities of the cells by undergoing these protein-protein interactions. Now, protein-protein interactions helps us to get a better understanding about different kinds of diseases and provides us informations about the development of different therapeutics. And from the structure of proteins, we can understand its function by determining the other molecule which can interact with it and as a consequence of its interaction. So multi-subunit protein, so one example or few examples includes your hemoglobin, then you have core RNA polymerase, small nuclear ribonucleoproteins, and we have the ribosome. Now, there are transient protein interactions which are strong and irreversible, and it readily undergoes changes in the oligomeric state. So the interaction of different protein kinases, protein phosphatases, then you have the glycosyl transferases, the acyl transferases, you have different proteases that basically cleave your proteins, interacts with their substrate proteins. So there are proteins for cellular growth, you have for performing cell cycle, metabolic pathways, and different feedback mechanisms or the signal transduction. Now, these interactions are basically very important in our lives as their disorder can lead to any fatal diseases like you have Alzheimer's or the Creutzfeldt-Jakob disease. So, the most studied or the well-known one common example of a protein-protein interaction is basically your actin and myosin while regulating the muscular contraction in our body. So the examples of different protein-protein interactions is based on your single signal transduction and which maintains the activity of the cell that is regulated by different extracellular signals. So from the structure you can see that there are different alpha helices and beta sheets, and you have the glutamic acid of collagen, and these two alpha helices are meant for giving informations about the signal transduction and all. Then you have the midas, and this is the glutamic acid from the collagen. Then you have different transport across membrane proteins, so a protein that may be carrying another protein. So you have that kind of proteins. Then protein interactions based on cellular metabolism. Like in many biosynthetic processes, enzymes interact with each other to produce small compounds or other macromolecules. And then, as I was speaking in the last slide about actin myosin, so you have the function of muscle contraction, like that act as your motor proteins, and they act as your molecular motors, and by binding to the actin enables the filament sliding. So basically, there are four, yes. So there are four examples of protein-protein interactions. So basically, you can tell that these are the functions of your protein-protein interactions. One is your signal transduction. Then you have transport across membranes. Then you have cell metabolism, and lastly is your muscle contraction. Okay, now there are different types of protein-protein interaction. The first one is on the basis of their composition. So when we categorize stuffs based on their composition, then we have the homooligomers. Like these are macromolecules, complexes having one type of protein subunits, like the protein-protein interaction that you observe in muscle contraction. Then you have the heterooligomers, that is made up of two different types of complexes having or multiple types of protein subunits, and this is the interaction between cytochrome oxidase and TRPC3, that is your transient receptor potential cation channels. Now, these two are on the basis of your composition, like the homooligomer and the heterooligomer. So, from the name, homooligomer means same type of protein complexes, and heterooligomer means multiple types of protein complexes. Then you have on the basis of duration. So, on the basis of duration, that is on the basis of duration means how they are interacting or for how long they are interacting. Okay, so when we categorize these protein-protein interaction based on duration, you have the stable interactions, that lasts for a long duration, and it's a very stable. So from the name, you can understand that it's a very stable interaction, or the interaction is very tight. So you call it a stable interaction. And there are numerous number of bonds or interactions that is occurring between the um, say amino acids of the two proteins. Okay, so there we call it a stable interaction, like that we observe for an hemoglobin structure. And transient interactions occur because occur due to a for a short period of time, and this is like your muscle contraction. Now here we have to study about the effects of protein interactions. So the protein interactions can basically alter the kinetic properties of proteins. These protein interactions are a common mechanism. Okay, so the protein interaction. So the effects of protein interaction includes your altering of the kinetic properties of proteins. Then the protein interactions are follow one common mechanism to allow the substrate channeling, and they can result in the formation of a new binding site. They have the capability to inactivate another protein, and they can change the specificity of a protein for its substrate. And from these interactions, you can identify several other interactions to understand the extent to which they can take place inside a cell and then determine the consequences of such an interaction. So, based on their composition, when we categorize, we have the homooligomers that I already spoke about it. So, homooligomers are macromolecule complexes constituted by only one type of protein subunit. So, the protein subunit's assembly is guided by the establishment of some non-covalent interactions in the quaternary structure of a protein, like you see for muscle contraction. Also, sometimes you see for some protein-protein interactions in muscle contraction. And there are several enzymes, carrier proteins, and transcriptional regulatory factors that carry out functions as your homo-oligomers. Followed by, you have the heterooligomers. That is, it is a combination of, it can be a combination of many types of proteins. So because you do not have only one specific type of protein that is basically interacting, so there can be numerous number of proteins that can interact with one another at a particular time. Like you have the hemoglobin. Okay, so the four subunits you have, and they are basically, you have the alpha chain, then you have two alpha chains and two beta chain. Okay, so that is, you can give an example of an heterooligomers. So heterologous proteins are involved in different cell signaling events. So protein-protein interactions between cytochrome oxidase and your TRPC3. Okay, that I mentioned before. Okay. Now, how do you categorize them based on their bonding abilities? So for that, you have the covalent type of a bonding and the non-covalent type of a bonding, right? So if it's a covalent type of a bonding, then it's the strongest association. That is, it can be the type of disulfide bonds or electron sharing. Okay, like you observe for the post-translational modifications, you have ubiquitinylation and the sumylation. Okay, and there is another kind of interaction like you have for non-covalent. So it is established during transient interactions by the combination of your weaker bonds. You have the hydrogen bonds, the ionic interactions, van der Waals forces, and the hydrophobic bonds. Those are the falls under the category of your non-covalent bonds, and the bonds involved between two proteins. You can call them as the non-covalent interactions, and like you have the disulfide bonds or the electron sharing, that you call as the covalent interactions or the covalent type of bonding between the two proteins. Now, let us study about ubiquitination. Ubiquitination. So, ubiquitination plays a role in the degradation of defective and superfluous proteins, and it's a single chain polypeptide. Now, ubiquitination or ubiquitinylation is an enzymatic post-translational modification in which you add a ubiquitin protein to a substrate protein. So how do you do that? So the steps involved over here are activation of the protein, then you conjugate the protein, and finally the ligation. So by ubiquitin activating enzymes, you perform the activation. Then you have the ubiquitin conjugating enzymes, the E2S, that performs the conjugation step. And finally, the ligation is done by ubiquitin ligases or E3S. Now, when you distinguish them on the basis of their interaction, you have the transient and the stable interactions. Like in the transient interactions, it lasts for a short period of time in a reversible manner. You have the G protein coupled receptors only transiently bind to the G proteins that is activated by the extracellular ligands. Then you have other stable interactions like the proteins that interact for a long period of time, taking part of permanent complexes as subunits, and they carry out functional or structural roles. For example, you have the cytochrome C. Now, stable interactions where you have the cytochrome C or the CCO complex, it is stabilized by a few electrostatic interactions between the long side chains within a small contact surface. And in contrast to other cytochrome C complexes, numerous water molecules are found in the long intermolecular span between cytochrome C and CCO. Now coming to another important topic, that is your protein domains. So interactions are only possible due to the structural domains within the protein. A protein domain is that part of the protein that has a conserved sequence over the generation, and a tertiary structure that can evolve, function, and exist independently of the rest of the protein chain. So these proteins hold structural domains that allow their interaction with and bind to specific sequences on other proteins. Now, phosphotyrosine containing motifs in the that you examples you have means what is the function of this phosphotyrosine containing motifs. For example, these are examples of proteins which carry this motif, like you have the activated receptors for growth factors, then you have the cytokines and the antigens, and in this category you have the SH2 domain and the PTB domains that also binds to unphosphorylated peptides. The second category is your phosphoserine or the threonine motifs. So you have these are important for recognizing the protein-protein interactions domain. You have the 1433 proteins, you have the FHA proteins, and the domains, and you have the WW domains which binds unphosphorylated peptides, and these are basically proline rich. Okay, so and the other one is your WD40 repeat domains. The third category is your acetylation of the lysine residues. So these are proteins who carry the histone motifs. So these recognize proteins and creates binding sites for the bromodomain. Then the methylation of the lysine residue. So acetylation is recognizing proteins and creating binding sites for the bromodomain. The lysine residues also carry the histone motifs, but they recognize proteins and creates binding site for the chromodomains. So acetylation, bromodomain, methylation, chromodomain. And there are other protein-protein interaction domains as well, which you can also study. And based on their function, if you tell, then you have the death domain, the death effector domain, the caspase recruitment domain, the BCL2 homology domain that plays important roles in apoptosis, You have the cold shock domain for the chromatin modifications and all. You have the F box, the HECT, that is homologous to the E6AP carboxyl terminus. You have the really interesting gene or the ring domain for proteolysis. For dimerization, you have the SAM or the sterile alpha motif domain. For vesicle trafficking, that is an important function in the cell. For that, you have the GYF, the snare, the VHS. And there are other protein-protein interaction domains which does not have a particular function, or their functions are not yet identified. You have the NK, ARM, WT40, and the LIM. Now, let us talk about a few of the domains where you have the SH2 domain or the SRC homology domain. So, its role is cellular communication, and it contains 2 alpha helices and 7 beta strands. It has a high affinity to phosphorylated tyrosine residues. It is known to identify a sequence of 3 to 6 amino acids within a peptide motif, and it represents the largest class of tyrosine recognition domains. So these are a few protein-protein interaction identification methods, which includes your yeast to hybrid system. So this is experimental in vivo. So you have the split ubiquitin system, the split lactamase, or the split galactosidase system, and you have the split yellow fluorescent protein system. Then for in vitro experimental assays, you have co-immunoprecipitation, tag fusion proteins, X-ray diffraction, biocore methods, then the phage display technique. And then in computationally or by in silico methods, you have the bind, dip, mint, and intact. Also, there are other protein-protein interaction assays which you can use to investigate these interactions. You have the immunoprecipitation, you have protein microarrays, you have the analytical ultracentrifugation technique. You can undergo dynamic and static light scattering experiments. Then you have the fluorescence spectroscopy. You have RETS, that is resonance energy transfer system. You can take the help of SPR, surface plasmon resonance, protein fragment complementation assays, and titration calorimetry. And two other prominent methods for investigating this protein-protein interaction is your yeast to hybrid system and affinity purification, which is coupled to mass spectrometry. Okay, so the yeast to hybrid system is used for testing for the physical interactions between two proteins. So it was first proven using the yeast Saccharomyces cerevisiae as a biological model by Fields and Song. So here you have the BET, a prey, and a reporter gene. So the reporter gene is basically your LAGZ reporter for your blue-white screening. The BET here is the protein that is fused to your DNA binding domain, and it is referred to as the BET. So this transcription factor like the GAL4, and the prey is basically attached to your activation domain. So you have two domains. One is the DNA binding domain or the DBD, and the other one is your activation domain. So the protein fused to the activation domain is your prey, and the bet is basically the one to which the protein is fused to the DNA binding domain. And the DNA binding domain will sit on the cognate binding site, and the activate when the bet catches the prey, and when the prey sits on the bet or comes in close interaction, or the bet and the prey interact, then the reporter gene is expressed. So, what is the basic principle of this is to hybrid screening method? Is that if you have a protein X and Y that interacts, then the DNA binding domain and the activation domain will combine to form a functional transcriptional activator, and the transcriptional activator will then proceed to transcribe the reported gene that is paired to its promoter. So here you have the bed plasmid and the prey plasmid. So if you can see that the bed plasmid has a gal binding domain, then you have the TRP1, you have the X, that is your protein, and then you can see it encodes for a particular gene, and then you have the hunter plasmid where you have the activation domain, where you have the gal4 activation domain, you have the Y, and the LEU2. So the bed plasmid, which is the protein of interest that is fused to gal4 binding domain, and the hunter plasmid, that is a potential binding partner that is fused to the gal4 activation domain. So the selection genes encodes for amino acids such as you have histidine, then leucine, and the tryptophan. So based on that, you can actually select the colonies or the plasmids or the cells that have taken up your plasmid or for which the protein-protein interaction is occurring. So this is totally a plasmid based system, and it's a plasmid based system, so only those cells that take up both of them and the interaction undergoes, they are going to survive, others will basically die. Okay, just 2 minutes. I am again restarting. Just 2 minutes. Okay. So the co-immunoprecipitation technique is also another type. So these are totally just because they're based on protein-protein interaction. So if you see, then you can understand even from the diagram that two proteins are basically interacting. So what is happening over here is that the protein-protein co-immunoprecipitation is a classic technique that is used for interactions between different proteins for the identification and validation. And the new binding partners or the binding affinities, the kinetics of the binding and the function of the target protein can be identified. Okay, so if you see from the diagram that it's a cell lysis by a non-ionic detergent, and then you incubate the cell with a particular kind of an antibody to which say y x binds, and then you remove the unbound proteins by washing, and then you can study this interaction by either Western blot or different mass spectrometric analysis. Okay. Now, the advantage of this co-immunoprecipitation technique actually here is that both the bait and the prey proteins are in the native conformation in the co-immunoprecipitation assay, and the interaction between the bait and the prey happens in vivo with little or no external influence. So, but there is a limitation of this technology. It is that is low affinity or the transient interaction between the proteins. So if it's say a weak interaction between the two proteins, then it cannot be identified. So the last one is your protein affinity chromatography. So the affinity chromatography is essentially a sample purification technique for the biological molecules such as your proteins. So it is a method for separating a mixture of proteins or nucleic acids by specific interactions of those molecules within a component known as a ligand, which is immobilized on a support. And if a mixture of proteins is passed over the column, one of the proteins bind to the ligand on the basis of specificity. Now, this is a sample purification technique for biological molecules such as your proteins. It is a method for separating a mixture of proteins or nucleic acids by specific interactions of those molecules within a component known as a ligand, which is immobilized on a support. Now, if a mixture of proteins is passed over the column, one of the proteins binds to the ligand on the basis of specificity and high affinity, like they fit in a lock and key model, and these proteins in a solution was through the column that were not able to bind to that particular ligand. So different fusion proteins, you have the glutathione transferase, you have the staphylococcus protein A and the maltose protein. So the applications of this protein-protein of this technique is to identify the novel protein-protein interactions, to characterize interactions already known to occur. And characterize interaction that is known to occur between say the protein domains. Then you need to understand the conditions of the interaction, and you have to know how mutation affects a protein's interaction with other proteins. So you can perform mutations on the interface or the amino acids, which you can check that they are involved in your interaction. So you can mutate that and check. And apart from that, there are other computational studies. There are databases. You have the deep, you have the bind PRD, you have the, there are other databases to have informations about the protein-protein interactions. They are collected through specialized biological databases. So these databases are divided based on primary databases, the meta databases, or the prediction databases. So the primary databases published protein-protein interactions proven to exist via small-scale, large-scale experimental methods like the biomolecular interaction network, bind. You have the BioGrid, HPRD. So they function differently, but ultimately their goal is the same, to gather information about the protein-protein interactions. So you have the primary databases, the meta database, and the prediction database. So the meta database, you have the primary and the original data. For example, the APID, the microbial, MPIDB, you have the PINA and the GPS prot, etc. And for the prediction databases, you have the protein-protein interaction prediction database, the PIPS, or I2D, string, unified human interactive, or the UniHI. You have other, so these use several techniques for the identification of protein-protein interactions. And these are databases, so they give information about the protein-protein interactions, or the PPIs. So in the conclusion, you have the protein-protein interaction methodologies that has been developed in the yeast methods and that are not suitable for all in the plant systems. You have other techniques like the co-immunoprecipitation, the affinity chromatography, and there are different databases. Also, you have the International Plant Proteomics Organization, the global initiative to develop and improve connections between plant proteomic researchers and related fields. And you understand that proteomic approaches are still challenging, and the therapeutic approaches to these protein-protein interactions has been a very hot topic for the researchers. So there are obviously certain therapeutics that needs to be developed against disease causing protein-protein interactions. So you have the several small molecules, antibiotics, you have antibodies, okay, and you have some kinds of peptides, be they linear or cyclic. So these act as different therapeutics for your protein-protein interactions, and they are going to be um, very helpful for the future. But it's a very what to say emerging field in the field of in research. Okay, now let us discuss with the questions. So the first question, which says which among the following is or are correct about your protein-protein interfaces? So if you see what are the options, the protein-protein interface is 50% polar. Polar residues in the interface are usually charged. The protein-protein interface is richer in amino acid aromatic residues. Protein-protein interfaces exhibit high shape and electrostatic complementarity. So if we see that the protein-protein interface is about 50% polar, that is correct, in the middle between the protein surface and the core region. So the polar residues in the interface are usually uncharged. Otherwise, it might cause, if there is a similar kind of a charge, it's basically going to affect it. So the first option is correct, like the protein-protein interface is 50% polar. This is wrong, it's not charged, it's uncharged. And the protein interfaces exhibit both shape and electrostatic complementarity to minimize their energy. And the protein interfaces are richer in aromatic residues, and histidine is also present to what to say, to enhance the interaction. So 1, 3 and 4 is correct. So 1, 3 and 4, yes. So C is the correct option. Okay, monoclonal antibodies act as modulator for targeting large protein-protein interfaces. They are used in screening recombinants, treatment of many cancers, and the diagnostic kits. So if you see that monoclonal antibodies are used in screening of recombinants, so because for their treatment in oncology and autoimmune diseases, and they are also can be used in different diagnostic kits. So basically, for the screening of recombinants, they can be used in the field of oncology and diagnostic kits. So all three are correct. So option E is the correct answer. Now, which among the following is or are correct about the SRC homology domain? So it is one of the two small protein binding domains found in the SRC oncoprotein. Then you have SRC is a non-receptor tyrosine kinase protein. It includes an SH2 domain, SH3 domain, and a tryptophan kinase domain. And the activated SRC regulates the activity of matrix metalloproteins 9. So if we just go back a few slides, and if you can check, see, you can understand that SRC homology domain is one of the two small protein binding domains that is found in the SRC oncoprotein. So this is correct. And it is a non-receptor tyrosine kinase proteins which is encoded in the human by the SRC gene. So 1 and 2 is correct. But it includes an SH2 domain, SH3 domain, that is correct, but not a tryptophan. It should be a tyrosine kinase domain. So this is wrong. It has to be a tyrosine kinase domain. And the activated SRC regulates the activity of matrix metalloproteins 9. So 1, 2 and 4 is the correct option. Now, which is not correct about the phosphotyrosine binding domains? So if you see that it interacts with sequences, containing the phosphotyrosine group, that is correct. The insulin receptor substrate contains these domains, that is also correct. It is situated towards the C terminus. That is not correct because it is situated towards the N terminus. And it is compact 7-stranded beta sandwich structure capped by an N-terminal helix. No, it has a 7-stranded beta sandwich structure that is capped by the C-terminal helix. So this is wrong, and this is wrong. Okay. Now you have to identify these domains. So the first for the identification of these domains, the first one is your beta sandwich structure. The second one is your double hairpin structure, and the third one is your double zinc finger motif. So the correct option is C. Beta sandwich structure 1, double hairpin structure 2, and the 3 one is your double zinc finger structure. Now next one, mimetics is used for. So peptide mimetics is used for identification of tight binding potential peptide sequence. That is correct. Modification of the peptide to enhance stability, uptake and delivery. That is also correct. Development of drug-like compounds from the existing peptide. So all three of the options are correct. So 1, 2 and 3, that is C. Now the next one. The staple proteins bind to estrogen receptor using a hydrocarbon link to stabilize them in the form of an what? Turn alpha helix random sheet beta sheet. So peptide inhibitors use a hydrocarbon link to stabilize them in the form of a alpha helix, that is known as your staple peptide. And shape complementarity is basically required for the protein to interact with your peptide over here. So you require an alpha helix to do that. Okay, now you have to find the incorrect options about the ACE inhibitors. So your ACE inhibitors. Okay, so your ACE inhibitors. So the angiotensin 2 is a hormone that is converted from angiotensin 1 by the enzyme ACE. Now the last two residues of angiotensin 1 are cut off by ACE, thereby producing angiotensin 2, and ACE is a low specificity carboxypeptidase. So number 3 is incorrect. So and 1 is incorrect. So therefore 1 and 3 are incorrect. This is correct, but the other two is low specificity. Now identify the protein from the following features. It belongs to a large family of molecular chaperones. Okay, so it belongs to chaperones. It has a key role in protein maturation, refolding, degradation, involved in protein folding in response to stressful conditions such as exposure to high temperature, and involved in folding of signal transduction proteins. So just because it says about molecular chaperones, it has to be some in the category of heat shock proteins. And the other one is exposure to high temperature. Okay, so just because it is exposure to high temperature, so based on 70 and 90, 90 would be the one. So heat shock proteins. Which of the following amino acids interact with gold nanoparticles by covalent bonding? So cysteine has a thiol group, and on the other hand, your gold has affinity towards sulfide. So cysteine can form a bond with your gold nanoparticle site, like the because it has a sulfur group, so it can form, uh, it can interact. So, but others cannot interact. So 2 is the right option. So choose the correct statements about the protein corona formed on nanoparticles. So the hard corona is formed by direct interaction of the protein with your nanoparticle, whereas the soft corona is formed by nanoparticle bound protein-protein interactions. Protein at the soft corona does not interact with the nanoparticles directly. So what is the option? Forms the protein-protein interaction, that is correct. And the hard corona is formed by nanoparticle protein interaction. So 1 and 3 are correct. And I guess we have the last one for the day. So we have to match the following: SRC, homology 3, SDM, we have LIM and the PDZ. So SRC3 domain binds with, yes, proline rich sequences. That is right. You have the SDM that binds to non-SAM binding containing proteins like RNA. So you have 1 and W, so 2, so 2W, so 1Y, 2W. Yes. So you have 1Y, 2W, you have 3, the limb is binds to helix loop motifs, that is right. And you have the last one, that is PTZ, that recognizes the ST QV motif. So D is the right option. Okay, so that is all from my portion, and I guess these questions will help you in solving your assignments and all, and this is going to be beneficial at some point of time. And you have an exam also coming up, so prepared. So this portion you might get a few questions from here, and that's going to be helpful for you people. Okay, that's all then. um, thank you all. We will meet in the last week lectures in the next week. Okay, thank you all.